My name is Bonnie Ertel, and I am a marine scientist who studies plastic pollution. I grew up in Greensboro, North Carolina, taking yearly trips to the North Carolina coast and absolutely fell in love with the beach from a really young age. I was lucky enough to have that exposure from a young age, which I do believe really shaped me to be the marine scientist that I am today. When I first went to college at UNC Chapel Hill, I didn't honestly know what I wanted to do. I got into the environmental science program, and they encouraged me to get some field experience going out into nature, doing science, and I signed up for a study abroad program called Sea Education Association, or SEA, or Sea Semester for short. And I did that program in 2016. This was my junior year. Still kind of not sure what I wanted to do with my career, what I wanted to be when I grew up. And that program really helped me figure it out. So I chose an independent research project as marine debris. Honestly, not really sure what that meant, but turns out that means plastic pollution. And in the Pacific Ocean, it's everywhere. You may have heard of the Great Pacific Garbage Patch, which is often quoted to be the size of Texas or even larger. And I think in people's mind, that brings to mind a picture of like a floating trash island, which is not exactly quite how it is. As we were sailing throughout the Pacific Ocean, directly through the Great Pacific Garbage Patch, you don't see big plastic items floating, but rather these small pieces of plastic pollution called microplastics. And we would take trawl nets going through the Great Pacific Garbage Patch and bring up just hundreds and hundreds of pieces, along with a lot of plankton and little fish and such, but the amount of plastic pollution that we were pulling that you can't even see with your naked eye was kind of mind-blowing, really concerning, and has inspired me to be the scientist that I am today. So when I got back from that study abroad, I did a semester down at UNC's Institute of Marine Science, where I did my honors thesis on marine debris in the North Carolina marshes, and also exposed fiddler crabs, which are the little one-armed crabs that run through the marsh. I exposed them to microplastics. And again, I was just completely mind blown at this issue of plastic pollution in our environment that had, up to that point, I was unaware. But once I started looking for it, you can see plastic everywhere. So after my undergraduate degree, I got my bachelor's in environmental science. I then did an internship with the Shaw Institute up in Blue Hill, Maine. And as a North Carolina girl, I really loved being able to get out of the state because, I did that study abroad in the Pacific Ocean where we sailed from Hawaii to Tahiti. And then I did my other study abroad on the coast of North Carolina, but to get out and see another environment up in the Gulf of Maine, where you would think it's really pristine waters. They have a little bit more regulations up there, and it's away from urban areas. But the amount of microplastics that I was sampling during my internship was just about as much as we were seeing out in the Pacific Ocean. And that really highlighted to me how these particles are everywhere. Microplastics have been found in every body of water we've looked at. They've been found in sediment, in air, in food, in tap water. They are what we like to call ubiquitous. They are everywhere. And in the last couple of years, they have gained a lot of attention within the research community as well as media outlets. Microplastics recently have been found in human blood. So that really excited me to see all of the research that is happening around microplastics. I didn't quite know that this was what I wanted to do when I first got into the world of environmental science. But now here I am living my life as a marine scientist, just making little Bonnie happy and proud, because I think I was maybe 8 years old when I went to SeaWorld, and I remember telling my mom, "Mommy, I want to do that. I want to be a marine scientist." And through one way or another, here I am today. After my internship, I reached out to John Weinstein at the Citadel, interested in doing some microplastics research, again, because I like this stuff. So he convinced me to go back and get my master's in biology from the Citadel, and in his lab, I helped work on several microplastics research projects. So the first one that I was part of was looking at microplastics in oysters. So what I did there was exposing oysters to microplastics in the laboratory to see what they took up and what they released, trying to understand how many microplastics we humans, if we were to consume oysters, how much are we at risk for consuming ourselves. Then my second project was looking at microplastics in the floodwater. So if you're from Charleston, you're probably familiar with the amount of tidal flooding, which will cover our streets. If you're not from Charleston, it's a major nuisance. We call it nuisance flooding because it can happen on even a sunny day. And our hypothesis was that the floodwater that's creeping up onto the streets could be taking particles and microplastics from the streets and depositing them directly into the creeks and into the harbor, which could then be taken up by different fish or plankton in the Charleston harbor. So first one was looking at oysters, then we were looking at floodwater, and my last project was quantifying marine debris along the southeastern United States coast. So we went to several beach locations and picked up what we could find on the beach face. So there we're not quite looking for microplastics, we're looking for bigger plastics. But that does really tie into microplastics because any big piece of plastic litter left on a beach is going to eventually break down into microplastics. I like to say that plastics don't go away, they break down, and they continue to break down for pretty much indefinitely. We don't know how long plastic pollution is going to stay on our planet. But every piece of plastic that has ever been created since the early 1900s is still on this planet in one form or another. Unless it's been unless it's been incinerated, which can release toxic fumes. But anyways, so those were our three projects looking at plastics in various forms and in various types of life. So after my master's degree, I decided to pursue a PhD. I'm working with Dr. Jeff Scott at the University of South Carolina, and I'm researching microplastics in dolphin intestines. So this project is in collaboration with NOAA and the Lowcountry Marine Mammal Network to obtain samples of dolphin intestines and look for microplastics. So dolphins are a great indicator of coastal environmental health. They are what we like to call sentinel species, which can help indicate changes in the environment, one of which being the increase of microplastic pollution in the environment. And looking at dolphins is a great, like I said, an indicator of coastal environmental health, and dolphins are mammals. We are mammals. We eat some of the same food. We eat some of the same fish that these dolphins are eating day in and day out. So for this project, I'm looking at a local population of Charleston dolphins who live pretty much strictly in the estuaries. They're eating, like I said, the same types of fish as we are, and they don't drink water. So any types of microplastics that are in the dolphin are likely coming from the fish they eat. Again, the dolphin eats the fish eats the plankton eats the plastic. So there is a chance that the amount of plastics in these dolphins is similar to what would be in humans. So I work with the Lowcountry Marine Mammal Network to get the intestines of locally stranded dolphins, and we'll do a dissection of that organ. I also am looking at stomachs some of the time when they're there. The samples are kind of limited, we get what we get from the animals, and depending on the degree of decomposition and other factors. So I want to look at dolphins. Like I said, they are sentinel species, indicators of coastal health, and they're also top predators. Other studies of marine mammals have shown that filter feeding species, such as the blue whale, can consume up to 5 to 10 million microplastics a day, which is quite a bit. But their feeding style is a little bit different, so they're just scooping up big masses of water, whereas these bottlenose dolphins are foraging more on the fish. But I think they're still interesting. And we have a local population here in Charleston, and we have samples dating back to 2016. So with that amount of samples and data, I can do a long-term analysis to see if the abundance of microplastics over time has increased in these dolphins as our coastal environment is changing. So now that you know a little bit about me and my background as a scientist, I would like to take you along and show you some of this process. All right, here are our facilities that we use at NOAA, working with Wayne McPhee. Let's come on and check it out. Here's where we do the microplastic. And then here is where we do the microplastic stuff. So now we're in the microplastics lab. This is where we're going to dissect and cut up the intestines to look for microplastics. And I am wearing this trendy yellow lab coat because microplastic fibers can be shed from our clothing all the time. So in order to not contaminate my sample, I'm wearing yellow because that way if I find yellow in my samples, I know that it's come from my lab coat. In terms of other personal protective equipment, I'm wearing gloves so that any bacteria or contamination is going to be on my gloves and then I can just take them off and dispose of them. I'm also going to put on a N95 mask to protect myself from the sample. So let's get to it. Of intestines. This is from an animal which was less than 5 feet, so still in the young juvenile age range. And once we've laid it all out and measured it all, I wanted to show you first the kind of anatomy of what we're looking at here. So this is the stomach. We have 3 chambers. First, the food's going to go through the esophagus into the forechamber. Then it goes into the fundic chamber. Lastly, the pyloric chamber. Then things are going to pass through the intestines, all 50 feet of them, until they reach the end and are excreted by the animal. So I'm going to divide the entire length of these intestines by 8, so that I'm processing them in 8 sections of equal length. And then, after I get one individual section, let's just say this is one section, I will cut lengthwise into the intestines and rinse anything that's contained within them to look for microplastics. So that is an overview of the process that I'm doing, and I know this is a little bit gross, but I think it's kind of fun. All right, so here are the contents of those intestines. So going from section 1 through 8, this is from the closest side to the animal and then further and further down the intestines. And this animal was pretty young, so what we mostly found was liquid, milky contents. And that makes sense because this animal would have been young, maybe eating a fish or two here and there, but mostly milky contents. But I bet I will still find microplastics. So the contents that you see here were rinsed through these sieves, and we went slow and steady to rinse the waste. And here's what we've got. Then each of these jars is going to go through another step to look for the microplastics. But for now, that's how we process the intestines. Once we get them from the necropsy into these jars, we do have a little bit more to go, but that's a different story for a different day. All right, so now we're in a different lab space. This is where I do a lot of the processing of the samples after we've done the dissection. So this is what we just worked on down in the other lab. And you can see it's kind of milky. There's maybe some microplastics in there, but to figure that out, what I'm gonna do is add potassium hydroxide. So potassium hydroxide, or KOH, is a strong base which is going to dissolve any of the natural or biological material. So I would say a lot of this is probably milk, which I would consider a biological material. When I add KOH to that, it's going to turn very clear, as you can see here. So this is from another sample which has had KOH added to it for about a week now, I add 10% KOH on hot plates to heat up the sample and dissolve any biological material. So once it's clear like this is really good, this is what I want. I'm going to pour it through this vacuum filtration system. So this is going to pull everything from the sample onto this type of filter. You can see here that it's nice and gridded, which is going to help me when I look under the microscope. I'll show you that in just a second. But this is the filter that everything is going to get sucked down onto. Then I put that filter into a glass petri dish. So if you have paid any attention, all along these steps that I've been processing things, I've used only glass. Glass is really great because it's not plastic. It's not going to be shedding any plastic materials to contaminate my sample. But in this glass petri dish, here is one example filter of a stomach that I looked at. And you see that black particle? That could be plastic. I am not too sure, but we will talk more about that in just a second, about how I'm going to be able to determine whether or not that piece is or is not plastic. Most of the plastics that I'm looking at are so small that you can't see them with your eyes. Again, they're microplastics. So to look at microplastics, what I mainly use is the microscope, which is over here. So kind of walking through all of the steps that we've digested the biological material, we've filtered it onto a filter. One of the last steps is to actually look at my sample under the microscope. So here I have a sample. Turn on my light, that would help. And again, you can see the gridded filter really helps me keep track of where I'm at. So what I would usually do is go along the tracks of this filter and pull out what I think are microplastics. So using my handy dandy forceps, which normal people call these tweezers, but here in the lab we call it a fancy word called forceps. And with my forceps, I'm going to very meticulously pull out anything that I think might be plastic. So here, perfect example, here is a clear fiber, which you may not be able to see on the screen, but I promise you it was there. Let's see if I can find another one. Ooh, here's a good one. So this is my probe, and here I'm pointing out a fiber that I think could be a microplastic. And what I'm gonna do is pull that plastic into a small jar, or vial, I guess would be the correct term. And then mark it on my sheet that I found one clear fiber. Then, the very last step of actually determining whether or not what I've put in my vials, if they are plastic, it's going to be a process called Raman. So Raman spectroscopy has no association to the noodles, but rather it's a way to chemically identify particles as either plastic or natural. So that fiber I just pulled out, it could be something like a cellulose fiber, which is a naturally occurring fiber. It could be cotton, or it could be polyethylene, polypropylene, polystyrene. It could be a poly, or a type of plastic polymer. So Raman spectroscopy, I will shoot lasers, and depending on how that laser beam is scattered off of the particle will tell me whether or not it's plastic. And I'm not going to get into the intense chemistry of it because sometimes, honestly, it blows my mind. But that, Raman, that is the very last step of this process. So now you've seen it all the way through from the guts to the spectroscopy, and that is how I look at a sample for microplastics. While microplastic pollution may be concerning because of the ubiquity and the amount that we find in the environment, there are things that you as an individual can do. So one of the most common types of microplastics I find in my samples are fibers. And these microfibers are being shed from our clothing. Every time you wash and dry your clothes, every cycle releases up to like 1,000 microplastics per cycle, probably even more than that, if I'm being honest. But a lot of them are treated by wastewater treatment plants, but some will still release into the environment. So one thing you can do is wash your clothes less often. If that shirt isn't all that dirty, wear it again, wash it later. And also looking for natural materials such as cotton or linen, anything that doesn't have poly in the name. So polyester, that's a plastic. It's actually the same kind of plastic that's used to make plastic water bottles. Another thing you can do is carry your own bag. Single-use plastic bags that are given to you by the grocery store are essentially unnecessary these days. If you can bring your own, please do it. I know sometimes you might forget it in the car, that's okay, but maybe reuse that bag. Another thing that you can do is reach out to local stores or your legislators and express to them that plastic pollution is concerning to you as a citizen. You have a voice, use it. And last thing that I would like to leave you with is reduce, reuse, recycle, refuse. Every item that you use as an individual is a vote towards, do you want to keep using plastic or do you not? You have every right to refuse the straw that's given to you by the waitress. You have every right to make those decisions in your everyday life.